So our last speaker for today is me. I introduce myself, I guess. I'm Jessica Carmen. I am a consultant with Poland Consulting, but I've spent my career in cell and gene therapies. I've worked for contract manufacturing, and I've worked for an enabling technology for gene delivery. And so Stefano had invited me to speak and sort of give an overview of the different delivery platforms that are available to people who are developing these products. And I really kept this theme of this Ready Player 2, this sort of second chance, second time, second attempt, second to market in mind when I pulled this together. I'd like to make a disclaimer also. In no way what I'm about to present is complete and thorough and the be-all, end-all, and there's nothing else beyond this. But I just pulled together some of my thoughts and some things I know that are available. And no doubt there's things being developed at this moment that we're not even aware of yet that could change. So I called this the next generation of gene therapy, a panoramic view. So I thought we'd start with a panoramic view of Miami, since we're all here in lovely Miami, and many of us don't live in a nice, warm, beautiful place like this. But, like I said, talk a little bit about the different tools. I put some slides up here where I kind of pulled some information together in a unique way. I've made my slides available. You're also welcome to take pictures if you are so inspired. So in thinking about this second time, second attempt, second to market, it made me think of the poem, The Road Less Traveled. And it made me think, it's not the road not traveled. It's okay if it's been traveled. It's okay that there's a path. The path is bumpy and rough and not easy to follow. Just because it's there doesn't mean that you shouldn't take it. And just because it's there doesn't mean it's useless and that it's not worth improving it, right? So it's not the road not taken. Yesterday was a talk all about the beauty and the curse of being first. It's difficult to get a product out the first time. There's so much unknown, right? And it's just obvious that there, regulatory considerations, technical considerations, scientific, those products are not going to be best in class. And everybody knows and acknowledges and accepts that. First products out aren't best in class, right? And my husband is an engineer, if anyone knows engineers and what engineers are like. They're not early adopters. They never want to take the first generation of a new technology because they know that there's going to be bugs. And so, you always go for, what's the next generation? That first generation is when you don't even know what you don't know when the first product comes out, right? And so, look at the Apple IIe. The things we all carry around in our pockets are much more powerful than these were. When I hear the Ready Player 2, I just keep hearing the Mario Kart song in my head. I don't know if everybody else is. So game on, right? So these products are on the market. That's cool, but there's companies that are in this space. They need to protect their IP by creating and innovating so that they have something to protect. And that's only better for the patients because that's making these products more diverse. It's making them more safe. It's making them lower cost. So all this innovation is only good for everybody, right? So, okay, great. What do we do? So here's the toolbox, okay? So here are the kind of common and maybe not so common tools that you may hear bandied about downstairs and at other conferences. You have your lentiviral vectors. Where am I going here? Okay, lentiviral vectors, adenoviral vectors, AAV, poration technologies, I say. And I'll talk a little bit more about that on the next slide. Your gene editing tools, so that's, ARCUS, as you mentioned, CRISPR, all those. I'll talk about that a little bit too. And then exosomes, which is something maybe people don't know about, which are these small little protrusions from the membrane of cells that cells put out, and they're just little bags of molecules. And so for some of these molecules that can play an important role, like cytokines and growth factors, they can be very labile. And trying to deliver these molecules, just putting it in someone's blood, they're never going to survive and they're never going to get where they need to go without a little bubble of protection around them, if you will. So that's what exosomes are, and they are starting to be used as therapeutic products. So, and then of course, so the expression, nothing is definite in biology, so I kind of bucketed them as whether they're permanent or transient, but then there's exceptions for some of them, but generally speaking. Poration technologies, there's nothing expression about that. That's pure delivery. We'll talk about that a little bit. And then exosomes as well, as I mentioned, the little bags of cells. And then, relatedly, is there an impact to the genome? For the exosome, because you can use an exosome to deliver gene editing tools or something along those lines, I think I would say it depends, maybe, right? And so, but is it a delivery? Is it a delivery method? Well, gene editing tools are not. They can do amazing things in the cell, but they have to get into the cell. They can't find their way in alone. They need a little help. And then, just for general interest, sort of payload size. And then with exosomes, of course, it depends because that's going to depend on the cell and the exosome and all sorts of factors. It depends. But what I think is kind of obvious, if you look at sort of the top half of the table, is viruses have got it all going on, right? They're your one-stop shop. They get it into the cell, and they can deliver it, and they can make a change. But not everything is one size fits all, right? And so I think that viruses play an important role, and there's a lot that's happening in the field that need viruses, but they're not always the answer. And so you have these poration type technologies, which is another way to get things into cells, and then you've got the gene editing tools, which may or may not, work well with virus. So the poration technology, pores, like little holes, right? So there's electroporation, which is the best known and best described and best deployed in the field, I would say. And so this is, you take the cells and you mix them in a conductive buffer with the molecules, and you give them a little buzz, and then the electricity causes the membrane to open temporarily, and whatever molecules are outside get in. Chemiporation is a chemical that will temporarily open the membrane of the cell. So again, if that's applied in combination with the molecule of interest, when the membrane is transiently opened, whatever's out will get in. Mechanoporation, it's a physical pressure or shear stress on the cells, maybe like a little hug. I think of it like a balloon. When you squeeze the balloon, you know that the membrane gets really thin, really, really, really thin at the biggest part of the balloon, right? And so what happens is at that point, the membrane is most porous and capable of taking up a molecule. And then there's also a sonoporation or ultrasonic waves, which actually the mechanism there is really what happens is when the wave is delivered, there's actually little tiny bubbles that occur at the surface of the cell, which also help open the membrane. So really what I'm talking about here is how do we just shove some stuff into a cell, right? We have these molecules outside, we want to get them in. And none of these technologies cause a permanent change to the cell. It's very transient. There's a pore for a little bit, whatever you want out is getting in, and then it's done. Electroporation is the only one of all of these that have been deployed in the clinic so far. So there are products, engineered cells on the market that have used electroporation to get the molecules into the cells. The others are not as far along in the development. And then is it scalable? With electroporation, we know that there is. There are technologies out there that actually will scale and make large numbers of cells, and you can electroporate them. The others, I put question marks because they're just too immature to know. But in theory, the technologies should be scalable as well. And I would expect that as years go on, we'll see that that is in fact true. So the gene editing tools, I didn't put ARCUS. So the CRISPR-Cas9, this is a cast of characters we've all heard of, right? So the CRISPR-Cas9, the zinc finger nucleases, the TALENs, Sleeping Beauty, and Piggyback, some of you may or may not have heard of before. And so I thought it would be worthwhile to mention sort of what these are made up of, because that really does impact how they can be delivered. And so they've all been deployed in the clinic, and they have all been demonstrated to be deliverable by the poration technology, and some of them have been demonstrated to be capable of being delivered by virus. The others may or may not be able to be delivered by virus, but to be honest, the Sleeping Beauty and the Piggyback, these are transposase systems, which means that you have a naked piece of DNA, a DNA plasmid, that's administered along with the enzyme that actually will do the cutting of the genome, which will allow the DNA to get inserted. So these tools here, the Sleeping Beauty and the Piggyback, need something like a poration technology to get into the cell. So they have got to get into the nucleus, and they can't get there by themselves. And so the next wave of ex vivo gene therapy. So I thought of a couple of interesting trends and product types that are sort of starting to emerge in the field. So CAR T co-administered with monoclonal antibodies. So there's a lot of clinical trials that are going on right now where they're doing a CAR T combined with a PD-1 checkpoint inhibitor. I actually moderated a panel almost 2 years ago now with Bruce Levine on the panel, and we were talking about this phenomenon of people administering this. And Bruce Levine said, if you could actually engineer the cell well, you actually don't need to co-administer with biologic. And he's right. And I think that some of these tools we were just talking about are really going to enable that potential to happen where you really don't need to co-administer. Bispecific CAR, so having a CAR that actually is recognizing 2 antigens at once, like a CD19 and a CD22 in one. And then to me, the third point, this CRISPR-mediated delivery of CAR by electroporation, this was a study that came out of a group at UCSF, and it was a publication that came out last July from Alex Morson's group, where they used CRISPR to deliver a CAR. So it was the first time where you really saw a truly non-viral, stable express CAR, and it really just made, I think, a lot of us in the field think about, wow, this is really starting to turn things on its ear, and you're starting to really see the finesse of the science developing and what people can do. And then, of course, the switches and the ability to turn things on and off and, turn something off once it gets into the patient or turn them on. And really it's allowing a more finer control. We think about, what Nicole was talking about this morning with the CRS. We know that the patient experience with these products isn't perfect, right? And the cytokine storm is important because it means that the product is working, but it's also, 100 and some odd fever for 6 days is pretty miserable, right? And scary and dangerous. And so a lot of these new engineering approaches that can be taken with CAR Ts are really going to help improve the patient experience with the product and also improve the safety and efficacy as well. So Ready Player 2, game on. We've got the toolbox. We've got academics out there describing new technologies right now that we can't even envision what they could be. And the rate at which these technologies are making it to the clinic is stunning. The rate at which the money is coming in is even more stunning, I would say. If you think about the fact that the first paper of Charpentier and Doudna describing CRISPR for its use to engineer human cells for therapeutic application was in 2012. And by 2015, it was already $1 billion business. The rate of growth of the field is just absolutely phenomenal. And so I think, the innovators keep innovating, and then the amazing science, as Pierluigi was saying this morning, the amazing talent and the amazing science is going to, the money will follow that. And we're going to see some really exciting products going to market. And it's a win-win. It's a win for the investors. They're going to get their money worth because we're going to see these products actually making it to the market. It's a win for patients because it's actually going to change lives and make things better for the patients. So game on. Thank you.